Good morning, all of you. Welcome you for this lecture series. So, in this lecture, I just want to talk about enzyme-linked immunosorbent assay, also called as ELISA technique for rapid detection of diseases. So, how this conventional technique is used to detect diseases rapidly and easily. That is the thing we are going to see in this lecture. I am Dr. Ashok Kumar, Associate Professor in School of Life Sciences, PS Abdur Rahman, Crescent Institute of Science and Technology, Vandalur, Chennai. So my email ID is given below. So if you have any queries, please do contact me. So first we'll see what is the definition of ELISA. So as I told earlier, the ELISA is abbreviated as the Enzyme-Linked Immunosorbent Assay. So it's an immunological assay which is commonly used to measure a variety of things. That includes, you can detect antibodies, antigens, proteins, glycoproteins, and also hormones in the biological samples. So based on these components, the diagnostic kits were developed. So those diagnostic kits include you can detect or diagnose HIV, pregnancy test that can also be done. The principle is ELISA, and also you can measure a variety of cytokines and soluble receptors in the cell supernatant or serum. So these are the biological fluids. Some of the biological fluids as well as cell supernatant, you can detect all these variety of things. So the discovery of ELISA technique was happened in the year 1971. A paper published by Peter Perlman and Eva Engvall from Stockholm University, Sweden. So these are the two people who published a paper about ELISA, how you can perform the ELISA, the principle behind it and everything. So let us see the principle of ELISA. What is the principle behind ELISA? So generally the basic principle of ELISA is antigen-antibody binding. So antigen and antibody, they bind together. Based on this simple principle, it helps to detect a variety of things. Okay. So as I told you earlier, the ELISA has been used for diagnostic tool in medicine. So it attempts to detect either qualitatively or quantify. You can quantify the sample in the presence of an antigen in the sample to see it as follows. So the antigens from the sample are attached to the surface. Then further a specific antibody which is raised against that antigen, that particular antigen, is applied over the surface so that it can bind to the antigen tightly. So that basically the antigen antibody, they bind very tightly. So this antibody is linked to an enzyme. So the antibody which you added, which is already linked with an enzyme. So the final step is you add specific substrate for that enzyme, where this substrate is colorless when you add, before you add. So after it reacts with an enzyme which is attached to the antibody, which will be cleaved by the enzyme, and the product will be the colored one. So which is detected, which produces a detectable signal, and most commonly this color change you're going to measure. This is a simple principle behind an ELISA. This is a sequential way how ELISA works. So before you go into the actual procedure of ELISA, so some of the terminologies you must know. So I have listed few terminologies. The first terminology is the antigen. So an antigen is nothing but any substance that causes your immune system to produce an antibody against it. So any substance that induces an immune reaction, that is called as antigen. So the antigen is variety of things, bacteria, viruses, peptides, all are foreign material to your body. So they raised antibody, our body raised antibody against those antigens. So next one is antibody. An antibody is also known as an immunoglobulin, so which is a large Y-shaped protein which is produced mainly by plasma cells, that is used by the immune system to neutralize these pathogens or this antigen which enters into your body. So next one is enzyme conjugate. So as I told you already, a specific enzyme which is attached to the antibody irreversibly. So the example of enzymes were given: ursodeoxyperoxidase, peroxidase, alkaline phosphatase, sorry, the alkaline phosphatase was repeated twice, and beta galactosidase. These are the enzymes that actually attach to the antibody. So next one is the chromogen or substrate. So this is a chemical which doesn't have a color. When it reacts to an enzyme which produces a color. So the specific enzymes have specific substrate. You can use trimethylbenzidine, orthophenyldiamine dichloride, peroxidase enzymes, they use these substrates. Para-nitrophenyl phosphate, this substrate which is specific for alkaline phosphatase. So a chromogen is nothing but, before you, it doesn't have any color. When you add it to the reaction, it reacts with an enzyme which produces a colored product. So keep in mind that. Next one is a stopping solution. So after you react, you have to stop the solution. So this process is nothing but to halt the enzyme substrate reaction. So many people commonly use sulfuric acid. So after you stop the reaction, to quantify the colored product, you have to read it in the spectrophotometry. Next we will see the types of ELISA. So generally ELISA, on the basis of binding structure between the antibody and antigen, it was classified into three types. The first type is indirect ELISA, and the second type is sandwich ELISA, and the third type is competitive ELISA. So we will see these three types of ELISA in detail with their procedure, how it works, and also what are the applications. Finally we will see what are the advantages and disadvantages. First we will look at the indirect ELISA. So what is indirect ELISA? So in indirect ELISA, an antibody can be detected or quantitatively determined by indirect ELISA. Okay. So in this technique, antigen is already coated in the microtiter well, which I showed you in the 96 well plate in the first introduction slide. So either serum or other body fluids or cell lysates, whatever the sample containing the primary antibody is added to the microtiter well and allowed to react with the coated antigen. Okay. So any free primary antibody is washed away. So you just wash after you coated the antibody. Okay. So the bound antibody to the antigen is detected by adding an enzyme conjugated secondary antibody that binds to the primary antibody. So the secondary antibody which has high affinity towards the primary antibody. So you coat that secondary antibody with an enzyme for the detection. So this unbound secondary antibody is then washed away and a specific substrate for the enzyme is added. So now the secondary antibody is added and which is already bound to the primary antibody. So secondary antibody contain an enzyme which is already attached. So when you add the substrate, so which this enzyme will react with the substrate and which start produce the color. So the amount of color of end products which is measured by means of a spectrophotometric plate reader that can measure an absorbance of all the wells in the 96 well plate. This is the overall protocol or overall way how an indirect ELISA works. So in the pictorial representative, you see the first figure. In the first box, the antigen is already coated So then again you have to wash it. So wash it means you use any type of even sodium chloride also you can use with a small detergent, 2 in 20 or something, you can use as a washing agent. So you wash it twice, then excess unbound antigen will be washed away, and then the next step after washing, you add specific antibody which is raised against the antigen. You add it, then this antibody will bind to the pre-coated antigen. So again you go for the washing step. After washing, you add the secondary antibody which is attached to the secondary antibody which is in the black color. You see the primary antibody is the blue color antibody, that Y-shaped figure, that is the primary antibody. So the red color dot is your antigen. So you add the secondary antibody which is already conjugated with an enzyme, that E denotes for conjugated with enzyme. So after you wash, the excess secondary antibody will be washed away, and then finally you add the substrate. The substrate will react to the enzyme and which gives you a colored product. Now you add the stopping solution, then you read it in the spectrophotometry which will give you a proper quantification of, you can quantify how much of your antigen is present in the well. So the procedure involves, first you have to coat the microtiter plate with an antigen, block all the unbound sites to prevent the false positive results. So only you open up where your antibody is going to bind in the antigen site. So that is the meaning of this blocking the unbound sites. Now you add the sample containing antibody. So the example is given here. They use the rabbit monoclonal antibody and then incubate the plate at 37 degrees Celsius. Then after the incubation time may vary for each experiment. Normally they incubate for 1 hour to 1.5 hours. Then wash the plate so that the unbound primary antibody will be washed away. So after this, you add the secondary antibody which is specific against the primary antibody which you used, which is already conjugated with an enzyme. The enzyme must be either you can use alkaline phosphatase conjugated, or you can use arthritis peroxidase conjugated one. After this secondary antibody step, you again wash the plate so that the unbound enzyme will be unbound secondary antibody conjugated with enzyme will be washed away. And then at the next step you add the substrate which is specific for the enzyme which is conjugated already. For these things you have to read the manual or the kit, whatever you are getting properly, what are the secondary antibody, what is the primary antibody, what is the secondary antibody, and what enzyme it is conjugated with, and what is the specific substrate you have to use for this. And then the reaction of the substrate with the enzyme to produce a colored product, this will be measured in a spectrophotometry. So I have given a curve here how we quantify the sample. For example, here I have given interferon gamma. So along with your unknown samples, we also take standard samples. For example, here I am going to quantify interferon gamma in a serum sample. So along with the unknown samples, I will take standard samples, a series of standard samples with different concentrations. So the treatments will be same. First I will coat the antigen, then I will coat the primary antibody, then the secondary antibody, then I will add the substrate, then I will measure it in the spectrophotometry. It is all for unknown samples, or the standard samples will be the same. So after I measure it in the spectrophotometry reader, so I will get the values, the optical density. Okay, so based on the optical density for standard, I will plot a curve. So on the x-axis I will use, you see the figure on the right side, on the x-axis I will use the concentration of the interferon gamma. On the y-axis I will use optical density. So based on the optical density received for each concentration, I will make a dot. So I will end up the concentrations, I will get a standard curve. So after this, I will take the unknown samples optical density. When I imply this with this standard curve, I will get the unknown volume, unknown concentration. This is how people quantify the sample. So what are the advantages of indirect ELISA? So it has high sensitivity because more than one labeled antibody is bound per antibody, and a variety of labeled secondary antibodies are available commercially. You can use based on the flexibility. So you can use any type of antibody, secondary antibody which is conjugated with different types of enzymes. You can use secondary antibody conjugated with alkaline phosphatase. You can use either secondary antibody conjugated with HRP, that is horse-radish peroxidase, or sometimes if it is available in your lab, you can use beta galactosidase coated secondary antibody. So whatever, based on the flexibility, you can use it. The maximum immunoreactivity is observed by the primary antibody, which is retained because of its not labeled. It is versatile because many primary antibodies can't be made in one species, and the same labeled secondary antibody can be used for detection. This is the one another advantage. And also it has flexibility because different primary detection antibodies can be used within the single labeled secondary antibody. Okay, and this will save your money. So cost savings, since because fewer labeled antibodies are required. Also different visualization markers can also be used with the same primary antibody. These are the advantages of indirect ELISA. So these also have couple of disadvantages. So the cross-reactivity might occur with the secondary antibody, resulting non-specific signal. The cross-reactivity means the antibody which is raised against rabbit, another antibody which is raised against rat may cross-react. So this will create a problem. This will create non-specific signal. So also this requires an extra incubation step that is required in the procedure. That is one of the disadvantages, but it is not a disadvantage. But compared to the other method, this is considered as a disadvantage. The second type of ELISA which we are going to see now was sandwich ELISA. So the name implies a sandwich. The antigen which is sandwiched in between two primary antibody, the same primary antibody, and when you add the secondary antibody which is already coated with the enzyme or conjugated with the enzyme, so this will bind to the capture antibody. Finally, when you add substrate, the enzyme which bound to the secondary antibody will react with the substrate and which will produce color. The color will be detected using spectrophotometer. So this is the basic setup of sandwich ELISA. So first what they do is antigen can be detected by antigen ELISA. This is the main use of sandwich ELISA. So in this technique what they do, the antibody is first coated in the microtiter plate. So the base, first they coat the primary antibody. A sample containing an antigen which is added to the well and allowed to react with the antibody which is already attached in the well or the plate, forming an antigen-antibody complex. So after the well which is washed, and excess sample and unnecessary things will be washed away, and second enzyme linked to the antibody specific for different epitope on the antigen is added and allowed to react with the bound antigen. Then after the unbound secondary antibody is removed by washing, finally the substrate is added and a plate which is hydrolyzed by an enzyme to form colored product. So in this picture we mentioned it carefully, right? So as a pictorial represent of sandwich ELISA. So this is the simplest way. I can tell you other way how people are doing. Our commercial kits, they follow the method which I am going to tell you a little later. So first we will see the sandwich ELISA. In the first monoclonal antibody which is coated in the well. First box you see an antibody which is already coated. Then you wash. This washing step will enable unbound antibodies will be washed away. So after that you add the sample which contain an antigen. It may be a serum, or it may be a plasma, or it may be any cell lysates. Then this antigen specific to the antibody which will bind to the antibody which is coated already there. So after that you again wash, and then you add the antibody, secondary antibody, which is already coated with the enzyme or conjugated with the enzyme. Then you again wash. Washing enables unbound secondary antibody will be washed away. So on the next one you add the substrate. So now the enzyme which is conjugated with the antibody will react with the substrate and which produce a colored product. This will be measured using spectrophotometer. So the second method, one more method, what most of the commercial kits available is, first they will coat the monoclonal antibody in the well, and then they add the sample. The antigen will bind to the already pre-coated antibody, and here the third step it is little difficult, where you add again one more primary antibody, the same antibody, where this primary antibody will go and bind to the antigen, form and sandwich. So after that you add the secondary antibody which is coated with the enzyme or conjugated with an enzyme. Then you add the substrate. So this is the way what the commercial companies provide. So they will provide you two primary antibodies. One is to coat the well, and the second is to bind with the antigen at the second stage. So what is the procedure of sandwich ELISA? First you prepare a surface which is known quantity of antibody is bound. Then add an antigen which containing a sample. It may be a plasma, it may be a serum, or it may be a cell lysate to the plate, and incubate the plate at 37 degrees Celsius. Wash the plate so that the unbound antigen will be washed away. And then the enzyme-linked antibodies, which are also specific to the antigen, that to be added at the second stage. Then again you incubate it to 37 degrees Celsius. Then again you go for wash the plate, and so the unbound enzyme-linked antibody, the second time you are adding the antibody, right, which will be washed away. So add substrate which is converted to the enzyme, converted by the enzyme to produce a colored product. So this reaction will be quantified by means of spectrophotometry. So what are the advantages of sandwich ELISA? So which has high specificity, since two antibodies are used in the antigen specifically captured and detected, and suitable for complex samples since the antigen does not require purification prior to measurement. Flexibility and sensitivity, since both direct and indirect detection method can be used. So the third type of ELISA is competitive ELISA. So in the competitive ELISA, what they do is, this test is generally to measure the concentration of antigen. All three methods are used to measure the antigen concentration, detection or measuring the concentration. In this test, what they do is, the antibody is first incubated in a solution with a sample containing antigen. The antigen-antibody mixture is then added to the microtiter well, which is coated with antigen already. The more the antigen present in the sample, the less the free antibody will be available to bind to the antigen coated well. After the well is washed, the enzyme conjugated secondary antibody and specific for isotope of the primary antibody is added to determine the amount of primary antibody bound to the well. The higher the concentration of antibody in the sample, the lower the absorbance. This is a basic principle. So in the pictorial represent, you see, first you incubate the antibody with antigen to be measured. So you add it to the mixture where your antigen-antibody mixture to the antigen coated well already. So here the antigen is already coated. You are adding both free antibody as well as the antigen coated antibody. So wash, and then finally you on the next step you add the secondary antibody, which is conjugated with the enzyme. Again you wash, then on the next step you add the substrate, and the enzyme will react to the substrate and which produce color. This will be measured in a spectrophotometer. So the procedure, how they follow the procedure means, first the antibody is incubated with sample containing antigen. So in a separate tube, an antigen-antibody complexes are formed. This complex will be added to the microtiter well, which is already pre-coated with the antigen. So wash the plate to remove unbound antibody. So the enzyme linked to the secondary antibody, which is specific to the primary antibody, is added at the next step. So wash the plate so that the unbound enzyme linked antibodies are removed. Add substrate which is converted to the enzyme into different kind of signals. Either it may be a fluorescent or it may be a normal signal, a color. So what are the advantages of this indirect ELISA? It is high specificity since 2 antibodies are used. It also high sensitivity because both direct and indirect detection method can be used here. Suitable for complex samples, again, which is since the antigen does not require purification prior to the measurement. So overall, what are the limitations of ELISA? So there is some limitations are there in ELISA. When you use, you have to carefully monitor these limitations before you use ELISA. So the enzyme mediated color change will react indefinitely over a sufficiently long period of time. The color strength will inaccurately reflect the amount of primary antibody present, so which yield false positive result. So you have to stop the reaction at the proper time, otherwise the color development will be higher and higher, so which provide a false positive result. To detect the given antibody or antigen, a known reciprocal antigen or a antibody must be generated. This is also one of the crucial limitation of ELISA. A non-specific binding of antibody or antigen to the plate will lead false high positive results. You cannot identify a non-specific binding of this antigen antibody. To overcome this, people increase the washing steps so that you remove the excess antibody or antigen, so that you avoid this limitation. So next comes to the applications of ELISA. So the application of ELISA, first presence of antigen or presence of antibody in a sample can be evaluated. That is the first application which we have seen even in the first slide. So determination of serum antibody concentration in a virus test. So we will see that in the HIV testing how they determine easily, and used in the food industry when detecting a potential food allergens. So that is one of the very important application of ELISA. Applied in diseases outbreaks, tracking a spread of diseases, example HIV, bird flu, common cold, cholera and STD, including the recent outbreak pandemic COVID-19 virus. Also they use a rapid kit test to detect this COVID-19. They use this rapid test kit. They developed a rapid test kits. We will see that in detail in the next slides. Okay, so based on this conventional method of ELISA, the rapid kits were developed to detect diseases, both communicable and non-communicable diseases, very easily and rapidly by using small kits. So we will see those things in detail. I'm going to give you 3 examples. So we will see that examples one by one. So the first thing is the pregnancy test kit. We all know that human chorionic gonadotropin is an hormone which is produced by the placenta after a fertilized egg is implanted there, which is detected in the, this particular hormone is detected in the urine when a particular woman is pregnant. So this hormone is available in the urine. So the presence of human chorionic gonadotrophin is detected in the pregnancy test. So the antibody which is already coated in a small membrane. So this test is very rapid and accurate. Anyone can use these test kits. No instruments are required to analyze these results, the results of these kits. So those are all very major advantages. Let's see how it works. So what they do is, in a small membrane, they put the antibody of human chorionic gonadotrophin, and then they'll also put two different sites. One is a test site. The test site, it contains HCG antibody, and the control site, it doesn't have an antibody. Okay. Oh, sorry, it also has antibody, but the HCG won't travel to the control site. So what they do is, in the reaction site, when you dip it in the urine, it attracts all the HCG. So there it also contains a dye and an enzyme. When you insert this into the test kit, where you see the test site, so where it contains a fixed antibody, where it traps the HCG. It also contains a dye that is called a substrate, where the enzyme and the substrate reacts and which produces color, which forms a small red color line. You see it in the picture. On the control side, what happens is, the fixed antibody which traps the free antibody, that reacts and which produces color. So that is control, means which proves the test is working. This is how the pregnancy test kit works, which is very cheaper, less than 100 rupees. So everyone is testing the pregnancy in home itself. After that, they will consult the physician. The second one is HIV TRI dot detection kit, so which is very rapid visual test kit based on the flow through technology which we have seen in the pregnancy test kit, which detects both HIV-1 and HIV-2. So this kit used the envelope antigens glycoprotein 41 C terminals of glycoprotein 120 for HIV-1 and glycoprotein 36 for HIV-2. This test is 100% sensitivity and 100% specificity as per the World Health Organization evaluations. The shelf life of this kit is 15 months when you store this kit in the 2 to 8 degrees Celsius, basically in the refrigerator. So you can visualize the results within 3 minutes. You can see whether you are positive to HIV-1 or 2 or negative. So no instrument is required again. So visually you can detect. So the principle behind this kit is an HIV antigen are immobilized in a porous immunofiltration membrane. It's similar like pregnancy test kit. A sample and reagent pass through the membrane and are absorbed into underlying absorbent. As the patient sample passes through the membrane, HIV antibodies, if it is present, bind to the immobilized antigens. So the conjugated binds of this Fc portion of HIV antibodies are give distinct pinkish purple dot. So this dot, which against to the white background, this dot indicates the person is positive. If there is no dot is formed, which is negative. The right side image is clearly given how it works. The third, then the recent development is COVID-19 rapid kits. So you all know that COVID-19 is a very pandemic, affects all over the world. So even some of the Indian companies also developed these kits. So the rapid test kit identifies the antibodies of SARS-CoV-2 virus that causes COVID-19. So at an accuracy of more than 95% within 10 minutes. So people use either serum or plasma or whole blood to test this kit. What really happens is this IgG and IgM antibody tests for COVID-19. Both this IgG and IgM are produced by humans in response to COVID-19 infection, right? So that's why they use this IgG and IgM. As such, the rapid test serves as an added tool for detecting COVID-19, indicate recent or prior infections. The kit details, the kit materials were given here. The principle is exactly similar to the HIV kits and the pregnancy test kit we have seen. Here also they have a control as well as the test site will be there. So in the test site, if it is positive, and also you have to get a line in the control whether the test is working, kind of internal control. So this helps patient to identify within 10 minutes he was infected to COVID-19 or not. So far we discussed too many details about ELISA. What is ELISA, some terminologies, and 3 different types of ELISA, what are the procedures involved, what is the advantages, what is the applications, what are the limitations, and some rapid test kits based on the ELISA. All these things we have discussed. So I used, these are the references. If you have any questions, my email address is given at the first slide. So you can contact me through email. I will respond to your questions. Thank you so much.